Okay, well, let's go ahead and get rolling for the day. So, announcements. Remember that Quiz 6 will be due. Is it Quiz 6? Yeah, it's Quiz 6. It's going to be due on Wednesday, so of next week. I posted the exam scores, and I put the exams in MRB 111 along with your problem sets as well. So problem set 1 is there. So go pick those up and look at them. Overall, I think that the exam went well, well enough anyway. It was a 73% average. If you were to take, and how I figured that out, if you're wondering, there's two, you should have 2 grade columns. One of them, we exam 1, the multiple choice, which was out of 60. And then the short answer, which was out of 30. Okay, so what I did is just took the average of those two columns and divided that by 90 and got the average of 73%. Okay, just so you know where that's coming from. Okay, so 73% is about what I'd expect, especially for the first exam of a class, and the material was not, I don't think, straightforward necessarily. It's a little bit demanding. Okay, so that kind of closes the book on exam 1. If you have questions on that, by all means come and see me, and you can ask your questions. Certainly ask questions of the TAs. I also posted a key on Canvas, so make sure you look at that. I had an exam recap module in Canvas. Look at the key in comparison to your answers. I didn't make a lot of notes in terms of why you missed points. In fact, I made no notes. Sometimes I would make a little note or something like that. So if you're really wondering, like, why did I miss some points, come and see me and I can give you a little bit more clarity on that. Okay? All right. So our riddle is really, really, really bad. What did the pickle say to the carrot? I know some of you are saying pickles don't talk. Okay. Besides that, but if they were to talk, a pickle would say to the carrot, "I'm kind of a big dill." So bad, right? And so here's the thing. I challenge you not to repeat that when it comes up, right? There's going to be something in the next week that's going to cause you, there's going to be some like uncomfortable silence or something like that, and that's what's going to come to your mind. And I'm sorry, right? Because that's going to make the uncomfortable silence worse. Because what follows afterwards is bad, okay? I'm just warning you. Okay, so on Wednesday, we started looking at what can be a very complex topic, and that is the regulation of glucose metabolism. I gave a couple of just summary slides, just like overall picture. This is what you could look at in regards to the regulation of glucose metabolism, and really only we're thinking about in this lecture is glycolysis and gluconeogenesis. We're going to expand that into a discussion on glycogen and glycogen regulation, and that is going to make it even more complex. All right, so we're not going to hit on every single aspect of this for sure. We're going to try and highlight some of the more important aspects. Certainly, insulin and glucagon and the effects that they will have on these pathways, specifically in the liver, is going to be very important. Okay, and we're going to look at that. We're also going to highlight some differences between liver, cardiac, and skeletal muscle as we move forward, though we won't get to that necessarily today. So we began then looking at the insulin signaling pathway. Actually, if I were to back up a couple of slides, we spent quite a bit of time on Wednesday on this slide looking at the events that will transpire after insulin binds to the tyrosine kinase receptor. And of course, I'm going to hold you accountable for those 7 steps, eventually, essentially leading to the phosphorylation and activation of AKT. And then AKT does stuff, and that's kind of where we were just a moment ago. AKT is kind of in the center here, and it's going to do lots of different things. And some of which we will look at this. This pathway is going to lead to, if you were to look at it, just like end results, for instance, it's going to lead to lipobiogenesis. I mean, that's an important topic later on as we get into metabolism of fats. Insulin is a signal to the liver to make fat. Okay, AKT can actually also, if you were to look at this, it can inhibit the production of proteins like PepsiK. I'll just circle this one. So this is going to down-regulate, in terms of genetic regulation, it's going to down-regulate the production of PepsiK in those cells. Gluconeogenesis flux is going to decrease in that response then. Okay, so like I said, there's lots of stuff that it can do. We're not going to go over all of them. Okay, now to highlight one more thing here, and this is where we ended with, and that would be P10. This would be an example of a way that this whole thing can be reversible. Okay, it's important to realize that insulin certainly, I mean, it has to be continually excreted for insulin signaling to continue to happen. And once insulin is no longer secreted, it'll be cleared by the bloodstream. However, its effects are covalent modifications of many things in the cell, not the least of which is the production of this PIP3, which is a covalent modification. Right? These are stable molecules, kinetically stable molecules, which means that if there's not some other enzyme that then converts them back to their original form prior to insulin signaling, then insulin signaling, despite the fact that insulin is not there, is going to continue. And that would be detrimental to many cell types, many tissue types. Specifically, I mean, if it's prolonged, it can actually lead to, right, unregulated cellular growth, and that would be not a good thing. Okay. So P10 is a phosphatase that then starts to take that PIP3 that accumulates during insulin signaling and breaks it down. Okay. Now that's P10, as I mentioned at the very end of Wednesday, is really unregulated. It's just there. It's just doing its thing. But as long as insulin signaling is there, the rate of PIP3 production is much greater than PIP3 breakdown. Okay, unless something happens to cause P10 to go up in expression, genetic expression. Okay, and there has been reports that that P10 can increase in genetic expression in response to inflammation pathways, and may be one of the puzzle pieces to understanding why type 2 diabetes is a problem. Okay, maybe one of the things that's causing some cell types to be what we call insulin resistant, something I'll talk about later on, and quite a bit in this class actually. Okay, all right, so that's just a review then of Wednesday, and what we wanted to go ahead and do now is not abandon, but to leave our discussion on insulin signaling and start talking about glucagon signaling, and look at that in some amount of detail, about the same amount of detail that we looked at insulin signaling, and then we'll start to apply this to glycolysis and gluconeogenesis. Okay. Before I do that, though, are there any questions on insulin signaling, any of the things that I said on Wednesday that may have come up? No? Okay. Let's do this. Let's talk about glucagon. Okay, glucagon, this is glucagon, actually. That's glucagon. This is the glucagon receptor. Okay, glucagon receptor, if you want to know this, okay, is what would be considered a G protein coupled receptor, GPCR, G protein coupled receptor. You may have encountered these in another class. Okay, I would imagine. These are really, really common. Okay, tyrosine kinase receptors are really common, which insulin is a tyrosine kinase receptor. G protein coupled receptors are extremely common. Okay, and they play a very large number of roles in lots of different cell types. To give you an idea, most of our sense of taste and smell and even sight comes from interactions of ligands and those types of things with G protein coupled receptors. And so there's quite literally thousands of these, okay, and types and that kind of thing. Okay, so glucagon is another version of these. All of these G protein coupled receptors are have seven membrane spanning alpha helices. Okay, the interaction then of some ligand with them will cause conformational changes or some different relationship with the receptor and this thing called the G protein. Okay. Now, before I go any further, let me just say this: we are going to look at glucagon. Okay, but we could, all we'd have to do, we could just cross this off and say epinephrine. And everything that I would say from this point on for glucagon or epinephrine would be identical. Virtually the same thing is going to happen when epinephrine binds to this or glucagon binds to this. Different cell types and different reasons that epinephrine would be commonly called adrenaline. Okay, adrenaline rush, that's what epinephrine is, the ligand that's released in a quote-unquote adrenaline rush. Okay, and that then affects what are called beta adrenergic receptors. It came out. I'm not going to try it again. Okay, sometimes you just trip over your own tongue. But anyway, it affects those receptors, which are also G protein coupled receptors. Okay, and the signaling pathways. The reason I'm bringing it up is because we'll talk about epinephrine and its effect on metabolism. But the signaling pathway is the same. Okay, past this point. Now, the G protein, let's get to this idea of what do we mean by G protein coupled receptor. G protein is a heterotrimeric protein that's embedded on the intracellular side of really any membrane that has a G protein coupled receptor. Obviously, it's coupled to these receptors. Okay. Heterotrimeric, 3 subunits, an alpha subunit. Okay, this doesn't project really great. Let me go ahead and go in on this. Alpha subunit, okay, and then there's a beta subunit, and then finally a gamma subunit, okay. And all G proteins have these. You'll notice in this slide it says G subscript S alpha, G subscript S beta, G subscript S delta. S stands for stimulatory. You can also have G proteins that would have GI alpha, I meaning inhibitory, okay. Glucagon or epinephrine lead to stimulation of things. Okay, there can be an inhibition of things as well. We won't encounter the inhibition aspects of this, but just to give you some idea. Okay, and when we say G protein, we don't mean there's one of these. There's lots of different isoforms of these. Okay, all right. So coming back to this, then let's go back to this view here. There we go. Right, so we could have just number 1, obviously glucagon binds to the receptor. Okay, so we have number 1 here, binds to the receptor, and then something happens. We won't get into the structural activities of this. If you took BC401, you have an idea of what might happen in terms of conformational changes. Okay, but a receptor changes activity of G protein, allowing GTP to bind red check minus alpha. Sorry, it's the alpha subunit. Okay, that's a typo. I give myself a red check minus for Friday. It's always a great way to start off class. Okay, right, but it's the alpha subunit that's going to do this. And so it will bind to GTP. Now, that's illustrated here, number 2. Okay, this guy here, okay, becomes this guy here. Okay. And you see now it has GTP bound to it. Okay. Now, again, I think I've said this in this class, but I'm not sure. It's really important, right? This, anytime a protein object is shining like that, that means it's been activated. Okay. Sunbeams apparently coming out of these poorly drawn objects, all right, means that they've been activated. Okay. So I'm sure that we're causing all manner of misconceptions when we do that within ourselves and within our students. But no matter, that's what we have. Okay. So once GTP binds to this alpha subunit, it will be activated, and it will then diffuse, and that's what you're kind of seeing here. Okay. It diffuses. I guess I should have highlighted this laterally on the membrane. Okay. Breaks its interaction with the beta and the gamma subunits and starts to diffuse laterally through the membrane. Okay. It will encounter a host of targets, one of which, and the one that we care about, is adenylyl cyclase. Okay? So this is adenylyl cyclase. Again, sunbeams. Okay? Sunbeams interacting only. Well, sunbeams only come about when it interacts with another sunbeam molecule, the GTP-bound alpha subunit of the G protein. Okay, and what happens then, adenylyl cyclase makes step number 4, cyclic AMP. Okay. That would be true, again, of glucagon signaling and epinephrine signaling. That would be true of some other types of signaling. We'll just leave it at that, though, for this class. Okay. So cyclic AMP is the result of this. Okay. Now, let me come down here, give you a little bit more, right, realistic view of what adenylyl cyclase is doing. It converts ATP, adenosine triphosphate, to the cyclic AMP molecule. And what we mean by cyclic AMP, let me go ahead and highlight this. Okay. The phosphate, the last phosphate on the adenosine molecule has a covalent bond to the 5' and the 3' carbon of the ribose. So it makes this, the reason it's called cyclic is because it makes this ring. Okay. And that's quote unquote cyclic AMP. Okay. And when adenylyl cyclase is active and activated by the G protein, it's going to make a lot of cyclic AMP. Okay. It's a fairly significant signal that goes through the cell. Now, cyclic AMP, of course, is going to do something. Okay, and we're going to look at what it's going to do on this next slide. So we've got step number 5 here. On the next slide, cyclic AMP binds to the regulatory subunit of pKa, causing release of activated catalytic subunit. I'll talk about that here in just a moment. But let me just put 5 here. Number 5, this is going to happen. Okay. I need to describe this figure to really describe what's happening with number 5. Okay. And so let's start at the top here, and let me go ahead and put a circle around something here. This right here is a protein. Everything that I just put that circle around is a protein commonly called pKa. Stands for protein kinase A. Okay? Protein kinase A. It's a tetramer. It has 2 copies of what we call regulatory subunits. And it has 2 copies of what would be considered the catalytic subunits. Okay? In this version here, they have R represented as a regulatory subunit, C represented as the catalytic subunit. And there's 2 copies, you see. Now, cyclic AMP that's being produced as a result of glucagon or epinephrine signaling will bind to that regulatory subunit, as mentioned in step 5 here, causing a conformational change in the regulatory subunit, then releasing, again, sunbeams, the catalytic subunits. So this would be C. Okay. And those are then activated. Right? So again, sunbeams mean active. I'll put a little plus in this. And then those go do stuff. Okay. That's the summary. Stuff. Okay. We'll look at some of the stuff here in a moment. And we'll certainly highlight some of the stuff as we move forward. Okay? Now, some of you are looking at that and saying, yeah, there's that green thing. What is that green thing? And you say, oh, it's AKAP. Ah, clearly. A kinase anchoring protein, if you're really curious about this. I don't want to spend a lot of time talking about A kinase anchoring protein. Looking into those, I didn't count them all, but there's a large number of isoforms of A kinase anchoring protein. These are found at different sites in a cell, okay? And they would be considered a scaffolding protein, okay? Something that hopefully you develop in your mind as a biochemist and as you move through cell biology and these types of things is that the cell has ways of having increasing local concentrations of certain activities by these scaffolding proteins. Okay? So what the scaffolding protein can do is it can concentrate a whole bunch of pKa in one point in the cell. And then it can concentrate targets of pKa within that same portion of the cell, so that when the signal from cyclic AMP comes, the catalytic subunit doesn't have to go diffusing through the entire cell to find its targets. Okay? Though it can, but it doesn't have to. Okay? Because that A kinase A anchoring protein can anchor not only pKa but other things in the surrounding environment. And it can put it on certain organelles and that kind of thing. It just depends on what A kinase A anchoring protein is being used. Okay, so. Now here's what's really fun about all of this. Because you have other coursework, okay, and you may have encountered pKa before and didn't even know it, or hopefully you did know it, but and the reason is because pKa can also be known as CAPK, cyclic AMP dependent protein kinase. Okay, so if you've seen the abbreviation cyclic or CAPK, then that's pKa. So in summary, pKa, aka CAPK, bound by aka AP. Okay? I see. I mean, only in biochemistry could I say something so stupid. Right? Some of you have been in the military and you're like, no, that's nothing based off of what you'd hear in the military. Okay? Okay. All right. So anyway. But you got to keep that in mind. Now, I will say this. I didn't mention this, but I could also make a very similar sentence. We could do this. AKT, AKA, PKB. Okay? So AKT was activated by, if you remember, insulin. AKT can also be called PKB. Okay? So protein kinase B. So the unfortunate thing about this is that as discoveries were made, people were working on the same protein and didn't realize it. And so they named them something different. And then as research developed and grew, then the fields merged and thought, oh, this is the same protein. Okay, and so they had two different nomenclatures for that same protein. And so then it's incumbent upon the rest of us to try and figure out the nomenclature. Anyway, okay? I will call it pKa exclusively, and I'll call it AKT exclusively. Questions? Super exciting stuff. Now, backwards. Let's go backwards just a little bit, okay? P10 was the means of reversing what we had talked about in regards to insulin signaling. This is reversible as well. And there are two aspects of it that are reversible. Okay, P10, by the way, is one means of reversing insulin signaling. We have to have some means of this. I want to talk about two of them. Okay, first of all, let's do this. There is a protein that's expressed much like P10. It's expressed as unregulated. It would be called constitutively expressed. And the protein is cyclic nucleotide phosphodiesterase, and it simply does this. It converts cyclic AMP back to just AMP, and you start to lose the signal. Okay. The cyclic nucleotide phosphodiesterase is consistently active. In the lack of epinephrine and glucagon signaling, right, virtually all the cyclic AMP will be converted back to AMP by this enzyme. Okay. In the presence of the signaling, just like in the presence of insulin signaling, the rate of this is not fast enough. There's not enough of this to keep up with the amount of cyclic AMP being produced. So essentially, the amount of the cyclic AMP that's produced by adenylyl cyclase in response to glucagon or epinephrine signaling overwhelms cyclic nucleotide phosphodiesterase. And that's how the signal is propagated. The other thing that we talk about in terms of reversibility is this. Okay, that G protein is a GTPase, which means that it will hydrolyze GTP, particularly the alpha subunit. Okay, so that GTP will remain GTP for only a small amount of time. And then the activity of the enzyme will actually convert it to GTP. At that point in time, it will diffuse back to, it will release adenylyl cyclase and diffuse around until it bumps into these other two subunits. If signaling is still present at the receptor, it will pick up another GTP and then diffuse back. And it will just go back and forth, okay, as long as the signal is present. Once the signal disappears, that GTPase activity will then essentially get rid of all of the GTP, and you'll have the GDP-bound alpha subunit that will then associate with the beta and gamma subunit and remain there. Okay. You have to have both of those things going on for this, okay? Because if it didn't have GTPase activity, even with this, cyclic AMP, even after you lost the glucagon signaling, or the epinephrine signaling, cyclic AMP would still overwhelm this. Or if you had only the GTPase activity, right, the cyclic AMP it's a stable molecule. So it would just remain in the cell and just keep having that effect, activating pKa. So you got to remove that too. Okay. All right. Questions? Yeah.
Are there any diseases associated with the phosphodiesterase not being expressed?
Okay, so the question is are there any disease states in which the phosphodiesterase would be not expressed or maybe expressed at a maybe lower level? And the answer is I don't know. So I think not expressed would be too drastic. Okay. And I think that it probably would lead to the death of the organism prior to even birth, I would think. But I'm not sure. So that's a guess. Considering how prevalent of a signal cyclic AMP is in so many different processes, okay. If you have sustained cyclic AMP signaling in lots and lots of different cells, lots and lots of different processes, it would be very harmful to the organism. Other questions that I can shoot in the dark at, make a guess? Okay. It wasn't quite shooting in the dark, but anyway. All right, now last thing here, let me just do this. I'll show you this slide for some appreciation. By the way, this slide looks very similar to the one I showed you with insulin. You can actually pull up these slides. If you go to this KEGG site, this genome, it's really, if you just search for KEGG biosignaling, you'll pull up the site. It's a wealth of information, okay? And you can pull up any really cell signaling pathway, metabolic pathway in it, and it looks something like this. And it'll give you pretty up-to-date information. You can click on each of those green boxes. It will tell you what that is, what it's doing, where it was discovered. It will link it to databases that will give you primary structures of it and all sorts of things. So really helpful website, especially if you're planning on going into research, in particular in cell biology, can be very useful. This is glucagon signaling pathway. Let me go ahead and highlight what we kind of went through. We did this, okay? We got to this, and we will do some of these things, right? So if you start looking at, you know, all of these things, this is what it's going to do, okay? Let me highlight one right now. Okay, pKa can activate something called CREB, okay, transcriptional factor that you probably encountered in some other courses, especially molecular genetics. CREB can activate once activated, and then the nucleus will lead to genetic expression of a host of genes, in particular PEP-CK. So glucagon is a signal to increase PEP-CK genetic expression, leading to gluconeogenesis. Insulin decreases that, okay? And so insulin will then remove an enzyme or reduce the amount of an enzyme found in the gluconeogenic pathway. Some of these other areas we'll get to later on, okay? Some of them we might not talk about at all. Other questions? Any thoughts, concerns about this? Hopes, dreams? No? Tears? It's that time of year, right? It's February, it's cold, miserable, and you're getting all your exam results back. Okay. All right, let me ask you a question. I've got a question for you. This will kind of tee us up for our next section here, okay? Glucagon signaling will do what? What do you think? I'll give you a minute 45. Look at that. It's going to upregulate glycolysis in the liver, downregulate glycolysis in the liver, upregulate glycolysis in the muscle, downregulate in the muscle, or a combination of those. Talk to your neighbor about it. What do you think? Just glucagon, big picture. What do you think is going to happen here? And then we'll start looking at details of this. On your marks, get set, go. All right? Minute 45. Talk to your neighbor about it. About 20 seconds left, so submit an answer if you haven't done so yet. About 5 seconds left, so give me that answer. Okay, let's check it out. There we go. All right, so 37%. Majority response say that B and D is correct. Downregulate glycolysis in the liver. I think they come out quite right. Okay, the liver, okay. And downregulate glycolysis in the muscle. That's 37% of you. 29% of you say it's going to upregulate glycolysis in the liver. Upregulate glycolysis in the muscle. That's our next best response. And then a variety of other answers in between. That's good. I'm glad that we're asking this question and we're going through this. So here's the reason I asked this question right now. Okay, we're going to head into now talking about tissue distribution of these glycolytic and gluconeogenic pathways and how they can be regulated within different aspects of different tissues. Okay, and so one of the things that I want to help develop in you is just start big, because we're going to get a lot of details. And if you can remember, like, big picture, the details will fall into place a lot easier. So big picture, when we think about glucagon, the big picture that you should be thinking of is that the pancreas has detected that the glucose in the blood is low, right? And in response then, it will excrete glucagon. Right? It will do the opposite for insulin. Okay? But glucagon has then been released. Now that, okay, is going to signal, it has really only two tissue types it affects. The liver and not the muscle, but adipose tissue. All right, so C and D are off the list, okay, because glucagon receptors are not found in the muscle. Now, epinephrine is, and we'll talk about what epinephrine can do there, okay? But epinephrine receptors, but not glucagon receptors, okay? So we're narrowed it down to now just A and B. Big picture here, okay? The liver, in response to glucagon signaling, needs to release glucose, which means that it better slow down glycolysis. Because if it doesn't slow down glycolysis, it will continually be using it and it will start to absorb glucose from the bloodstream, which is the exact opposite of what it needs to be doing. Okay? So the correct answer here is B, 12% of you. I mean, Some of you are down here. You're like, yeah, I knew it kind of. I just forgot. It was in the muscle. Okay, you got me. Good job. You tricked me again, you dirty. Just leave it at that, right? Okay. I know. The honeymoon phase is over. It's not funny anymore. Anyway, okay, but that's the big picture, okay? So when we think about this, that's how I would encourage you to start to work your way through this, all right? Start big. This is our signal. This is what it should be telling these tissues to be doing, okay? And move from there into the details, and then the details will fall in place, okay? So let's go ahead and look at this here. And some of these details. And we're going to start by looking at the liver. However, these next two slides, I'm going to go ahead and this is not really a mistake, but I'm just going to cross this off because everything on these next two slides is true of any of the tissue types that we're going to talk about. Okay, just the next two slides. And then we'll apply this to the liver, cardiac tissues, and skeletal muscle. Okay. All right, but when we start to consider regulation of glycolysis and gluconeogenesis, or really PFK1 and FBPase1, one of the main means of regulating these is through this molecule fructose 2,6-bisphosphate. And you think to yourself, that sounds really familiar, like really similar to fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate is the product of PFK1, which catalyzes an irreversible step and also the third step in the glycolytic pathway. It's great job. I'm glad if you worked your way through that, that's good, okay? But F26BP is not F16BP, okay? So fructose 2,6-bisphosphate is this molecule down here, okay? And it's got this phosphate on the second carbon rather than the first carbon, okay? So that's the molecule here, okay? So really, we're looking at here, this phosphate is on number 2 carbon. So this is the number 2 carbon. Okay. Fructose 1,6-bisphosphate would put it on this carbon. Okay. And you think to yourself, that's a really small difference. Exactly. Okay, but that's what we see in biology. Okay, very small differences make very large differences. Okay, so going back to this molecule, fructose 2,6-bisphosphate is going to activate the enzyme PFK1, right? And this is glycolysis. Okay, as a glycolytic enzyme, irreversible step. Okay, on to fructose 1,6-bisphosphate, okay. And it will inhibit. Okay, minus FBPase1. That would be downregulating essentially a gluconeogenic enzyme. Okay. Reciprocal. This is through allosteric activation or inhibition. It's doing one or the other on one of these enzymes. Okay, and that's true in all tissue types that express PFK1 and FBPase1. If fructose 2,6-bisphosphate is present, PFK1 will go up in activity, FBPase1 will go down in activity. Now, to give you an idea, this is really sensitive, okay? And I'll just give you this. This is relative velocity. This is of PFK1, right? And what you see here, these are different concentrations of the molecule fructose 2,6-bisphosphate. And you can see at 1 micromolar fructose 2,6-bisphosphate, we have a drastic difference in activity of PFK1. Now, in terms of concentration, 1 micromolar is really low concentration. So this is a really sensitive, okay, effect. So keep that in mind, okay? Fructose 2,6-bisphosphate, very sensitive in terms of activating PFK1 and inhibiting FBPase1. I don't have a graph like this for FBPase1, okay, but it's sensitive as well. Okay, and it will go in the opposite direction though. Right. Now the other thing here, again, for all tissues, right, and this is going to be really important, and that is how does then fructose 2,6-bisphosphate get made and broken down? Okay, again, reversibility is very important in this. Okay, and there is an enzyme, and that's what we see over here, this guy, okay, that we call, it's a bifunctional enzyme, and one of the domains, it has two domains, is the PFK2 domain. And you think, that sounds really familiar, like very similar to PFK1. Yeah, I know. All right, so that's, again, another unfortunate thing. But this is PFK2, all right? We'll call it the kinase domain. And then below it would be FBPase 2. Okay, not FBPase 1, but FBPase 2, and that's the phosphatase domain. Okay, and this, the kinase domain, will do this. It will take fructose 6-phosphate and make fructose 2,6-bisphosphate. And the phosphatase domain will take fructose 2,6-bisphosphate and break it down to fructose 6-phosphate. And they're in the exact same, this is one primary structure, so they're in the exact same protein. This is not a quaternary structure, this is actually a tertiary structure of a protein, two distinct domains. And the domains do the exact opposite of each other. One of them makes fructose 2,6-bisphosphate, the other one breaks it down. Now, if you're sitting there thinking to yourself, well, if they're both active at the same time, then it's just going to be spinning its wheels worthlessly. And that will be true, okay? So these are regulated with respect to one another in the same enzyme. Okay? When this is active, the bottom half of this, the phosphatase domain, if it's active, so I'll put positive, this will lead to increased gluconeogenesis, really increased FBPase1 activity. Okay? And the reason is because going back here, you're removing an inhibitor of FBPase1. It will also decrease PFK1 activity. Because you're removing a potent activator of PFK1. Up here, okay, if this is active, the kinase domain, then that is going to lead to increased glycolysis. And by that we mean increased PFK1 activity and decreased FBPase1. And you have to keep these things straight. And this is what I'm talking about, the details. It can be pretty hairy. Okay. All right, now, questions on that? No? Okay. Let's do this. Okay. Here is, and now we are going to the liver. Okay. So what I said here, that's going to be true in the three tissue types that we're going to talk about. Okay. Now, liver. Okay. This is that same protein. Okay. So we have a kinase domain here. So this is the PFK2 / FBPase2 protein, which is shown in a kind of a box form. And in the liver, the isoform that we find in the liver has on the N-terminus, a phosphorylation site. And when it's phosphorylated, the way it's shown here, okay, the kinase domain will be inhibited. So this inhibits kinase domain. I can't remember. I'm pretty sure it's a serine side chain that gets phosphorylated. I can't remember exactly. It's on the very N-terminal domain of this, but it will inhibit the kinase domain. And if we have it like this, going back to our universal, right, sunbeams, there we go. Okay. Right? So let's do this now, okay? So that's true in the liver. Let's go one step further and look at this slide here. Glucagon signaling, which leads to increased cyclic AMP production, activates the protein. See, and this is, the book uses cyclic AMP dependent protein kinase. So we'll just do this. That's PKA, right? So all of this stuff here is what we talked about at the beginning of class. Okay. So glucagon signaling activates cyclic AMP, which then activates PKA, which then PKA, one of its targets is this bifunctional protein. It will phosphorylate the kinase domain, that's what it's showing here, of PFK2, inactivating it, and again, some beams activate this. That will then lead to this fructose 2,6-bisphosphate breakdown. Oops, that's not supposed to be a G, it's supposed to be a 6. There we go. And that will then lead to, of course, inhibition of glycolysis and stimulation of gluconeogenesis. Now remember, go back to the question that I asked you. Okay. What does glucagon signaling do? And we said that it should inhibit glycolysis, okay, which is doing here by removing fructose 2,6-bisphosphate. It'll also stimulate gluconeogenesis at the same time. Okay. That means the liver is now going to export glucose. Okay. Yeah?
Would the kinase from a different one of these proteins inhibit the other one by phosphorylating it?
Okay, so the question is, would a kinase from one of these other proteins,
Another one of the same kind.
another one of this, PKA? Or another one of these guys?
Those guys.
Oh, could they phosphorylate themselves? No. No. So PFK2 would be the kinase domain of that. It can only phosphorylate fructose 6-phosphate. That's a substrate. They can't do big things. Yeah. Now here's the other half of this. Think big picture, insulin. We'll end with this. Big picture. The pancreas excretes insulin because the pancreas has recognized, okay, and through means that we'll talk about, recognized that blood glucose levels are high. That means that the tissues to start to take in glucose, including the liver. The insulin leads to this activation of this, okay, phosphoprotein phosphatase, right? And that phosphoprotein phosphatase will remove that same phosphate, activating PFK2, which means that the kinase domain will make fructose 2,6-bisphosphate. And that will then stimulate glycolysis and inhibit gluconeogenesis. In that case, then, the liver, because glycolysis is stimulated, will start to pull down glucose concentrations in the cell. GLUT2 will equilibrate that, right? And glucose will start to come into the liver. Okay? All right. And that's what the liver does. Let's stop right there. It gets really fun on Monday. Okay? Really fun. So, all right, so we'll see you on Monday. All right? And at this point in time, probably wait on the quiz. Okay? Quiz 6. We're not quite there in terms of what we need to cover for that. Okay?